Okay, this section is our start of the study of cells. We'll discuss the components of the cell, which is information from Chapter 2, and also how the membrane around the cell functions, which is information from Chapter 3. The cell is the fundamental structural unit of life on Earth, and it's separated from its environment by a cell membrane, and in some cases also a cell wall. Organisms can be single-celled or composed of only one cell, or they can be multicellular and composed of many cells. There are two general cell types. The first type is the prokaryotic cell. An example of that would be bacteria. The prokaryotic cell is much smaller than the eukaryotic cell, and there is no nucleus. There is a cell wall and also a cell membrane that encloses the prokaryotic cell. Eukaryotic cells are larger cells. Some examples of that would be protozoa, plant cells, fungi, and animal cells. They do have a nucleus, as well as other organelles that are enclosed by membranes, and a cell membrane encloses the cell. Some eukaryotic cells will have a cell wall. Plant cells are an example of that, and others do not. So animal cells do not have cell walls. Plasma membranes enclose every cell, so either a eukaryotic cell or a prokaryotic cell, they are going to be enclosed by a plasma membrane. And a plasma membrane has two layers of phospholipids, so the membrane is actually sometimes called a phospholipid bilayer. The polar heads are hydrophilic, and so they orient outwards to the external environment as well as inwards to the cell environment, both of which are usually composed of water. The hydrophobic tails orient towards each other between the two layers of phospholipids. Proteins are found studded throughout the membrane. The proteins and the phospholipids can move around laterally, so side to side. So this idea of the cell membrane is actually called the fluid mosaic model. It's fluid because the components move around. The mosaic part has to do with the proteins that are studded throughout. The proteins in the membrane serve as transport channels, as well as signaling proteins to send messengers to other cells. Due to the polar heads and the hydrophobic tails, the phospholipid bilayer is selectively permeable. So what this means is that some particles can cross the membrane very easily on their own. Examples of this would be very small hydrophobic molecules, such as oxygen and carbon dioxide. Some particles can cross on their own a little bit more slowly, like small uncharged polar molecules such as water, and other molecules are too big or are too charged to pass through the membrane on their own. Diffusion is the flowing of particles from an area of high concentration to an area of low concentration. So they go from an area where they are crowded and there's too much of them to an area that's open where they can spread about until an equilibrium is reached, where the particles are equal on either side of the membrane. In terms of the cell membrane, only very small particles can diffuse across on their own. Slightly larger particles can diffuse with the help of protein channels in the membrane. The protein channel might be nonspecific, allowing any particles of a certain size across, or it might be specific to a certain molecule, kind of fitting like a lock and key. This type of transport across the membrane is called facilitated diffusion. The movement of water across the membrane is called osmosis. Osmosis moves water from an area of high water concentration to an area of low water concentration until it's balanced, until it reaches an equilibrium. It can occur through regular diffusion across the membrane or through special proteins called aquaporins. If a cell is placed in a salt solution, the water will actually diffuse out of the cell through osmosis, causing the cell to shrivel up. Diffusion, facilitated diffusion, and osmosis are all examples of passive transport. So that's a type of transport across a membrane that does not require energy. Active transport, on the other hand, does require energy. In some situations, the cell needs to maintain a specific concentration of molecules inside the cell versus outside the cell that is not in equilibrium. So to maintain the concentration, cells will use proteins powered by ATP to move substances up the concentration gradient. Since ATP is used, this method of transport requires energy. Exocytosis and endocytosis involve the movement of large particles that can't cross the membrane through normal protein transports or diffusion. In exocytosis, a membrane-bound vesicle fuses with the plasma membrane, expelling its contents out of the cell. In endocytosis, a new vesicle forms around the particles as a result of the plasma membrane enclosing those particles and then pinching off. In addition to the cell membrane, eukaryotic cells also have a membrane-bound organelles, many membrane-bound organelles inside the cell. Organelles are like little organs. They each have a purpose in the cell, just like you each have a purpose in your body. This is a look at the inside of a generic animal cell. The nucleus in the center houses the majority of the DNA and RNA found in the cell. The dark spot in the center of the nucleus is called the nucleolus, and it is made up of tightly packed DNA molecules in the form of chromatin. This is where ribosomes are produced. So nuclear pores are holes in the nuclear envelope or membrane that allow RNA and ribos to exit as needed. The mitochondria are the powerhouses of the cell. Through the process of cellular respiration, they use glucose and oxygen to make ATP, which is the energy currency of the cell. Lysosomes are the disposal units of the cell. They chew up unnecessary wastes with digestive enzymes so that the components can be reused by the cell for other purposes. The small dots are ribosomes, and these are protein factories. Ribosomes can float free in the cytoplasm, the fluid in the cell, or they can be attached to the rough endoplasmic reticulum. The rough ER is a network of membranes that starts at the nuclear envelope and extends outward and is used for protein synthesis. Smooth ER, or smooth endoplasmic reticulum, doesn't have any ribosomes attached to it and is usually involved in lipid synthesis. The Golgi apparatus looks like a series of flattened, stacked pancakes. It takes proteins that the rough ER has produced and modifies and packages them for shipping outside the cell. Centrioles are involved in separating genetic information during cell division. The cytoskeleton is made up of protein fibers, such as microtubules, that give shape to the cell and act as railroad tracks for organelles moving throughout the cell. A plant cell is very similar to an animal cell, except that it also has a cell wall for added support. There is usually a large storage component for water in the center of the cell, and that's called the central vacuole. Plants have a chloroplast, or many chloroplasts, in addition to mitochondria. Chloroplasts are the location for the process of photosynthesis, where solar energy is converted into chemical energy in the form of simple sugars. Alright, so that ends our tour of the cell and the brief discussion on the cell membrane. Hopefully that was helpful. Please contact me via email or text message me on Skype if you have any questions about this.